What I'm going to tell you about in my approach is, and when I talk about abuse and use of antibodies in the lab and the clinic, is really, when I mean the clinic, I mean not treatment antibodies, but rather antibodies used for diagnostic purposes, either to make a diagnosis or to measure a protein or assess the presence of a protein, which is ultimately going to lead to a therapy, which may be a $100,000 or $200,000 a year therapy. So small test, inexpensive tests lead to a huge burden to the healthcare system. These are my disclosures. I don't know, it's not a CME talk, so I probably didn't need to show this, but in the medical world, I'm not allowed to speak without showing this. So this paper was, this is an editorial or an essay written by Manya Baker talking about the problems with antibodies. And my lab, as it were, was the poster child for this problem of an antibody that we purchased and were about to produce into a clinical test, had my best RO1 score ever of first percentile to produce this clinical test. And when we went back to buy the same antibody from the same vendor, it did not, it produced completely different results on the same tissue. And so that entire 2 RO1s worth of funding went down the drain with this process, and that's what was discussed in this paper. But the problem really was that the bad antibodies were that they cross-react, they're variable, or used in the wrong applications. To hear the full gory details, you can read her essay. But this is not a problem of an individual. This is a problem of a field. And I think Andrew's presentation was a great introduction, and I'll try to shorten this part. But I just wanted to talk about a survey that Science did to look at reproducibility. And they had 300 respondents to this survey, and 98% of the researchers said they're performing more rigorous quality controls, increasing sample sizes, and ensuring thorough documentation, but still less than 1 in 4 are able to reproduce what's going on in other labs. And what's that problem? So do you consider irreproducibility a problem? Yes, most of them do. And in the last year, have you been able to reproduce work from other labs? Only about 20% of the time. And what's causing the problem? Animal models and antibodies. So their survey, and I think other surveys that we'll hear about later, really suggest that antibodies are a key part of the reproducibility problem in science. You already saw this picture from work where the antibody reproducibility was assessed by Matthias Uhlin and his group, and found that that of 5,000 and some antibodies obtained from external sources, very few were reproducible, in fact, in the 35 to 40% range. So this is what I want to talk to you about over the next 25 minutes or so. I'm going to give you some examples of antibody abuse or failure or validation. We'll go through that part quickly, and then I'll tell you about what we do to try to combat this problem. And right now it's what we do or what you do. It's not what will be done by your vendor. Perhaps eventually it will be what's done by your vendor or by your government or by some other agency. But right now I'm going to talk about problems with sensitivity, specificity, and how to do validation, and then who's responsible for it. The problems that I'll discuss are the EGFR, Erbitux, estrogen receptor, and PD-L1, all clinical tests as a pathologist that performs these tests. So this is a little bit old now. These are antibodies that we tested for EGFR because we were interested in finding a companion diagnostic test to Erbitux quite a few years ago at this point, but still relevant for cetuximab. And these antibodies all bind different regions of EGFR, and these are the vendors and the various recommended retrieval sources. And this is comparing each to each other. So we've done quantitative comparatives of each, and you can see that most of these antibodies have regressions that are completely unrelated and not significant with respect to how the antibody recalls. Yet all 5 of these antibodies were used in different pathology labs around the country to test for EGFR expression. So you can see maybe it was partly explained because they bind to different domains of the antibody, and in fact 2 of them actually regress. They have an R-squared of 0.61, which isn't too bad, but not too great, suggesting this might have been why we've had such problems with EGFR. And in fact, if you looked at these different antibodies in a population of patients, some are not prognostic at all, and some actually are close to statistically significantly prognostic, or this one, as in fact shows statistically significant prognosis. This is summarized in this paper, but more seriously summarized by CAP. So CAP is the College of American Pathologists, and we're the organization that tests CLIA labs. That is, we provide surveys to laboratories, and the laboratories have to complete the survey. Then other members of our society go around and check that they've completed the survey and that they got the answers right. And if they didn't get the answers right, they get a chance to get them right the second time. And if they don't get that answer right the second time, then they lose their CLIA lab certification. So it's a little bit of an oversimplification, but that's how we currently govern our laboratory system in the US. And so this is actually from a CAP survey result for EGFR. Once we realized after approval of the drug, we needed to test for EGFR, we started testing it. And in 2005, only 4 of 10 test cases, so we sent out 10 real test cases, and only 4 of those cases showed 90% concordance in 70 labs. That means in 70 labs around the country that wanted to offer this test for sale, that wanted to bill for this test, they all tested it, and they couldn't agree amongst themselves. And at CAP, we don't have a gold standard. Our standard is concordance. And so we thought, well, we'll fix this. We'll take out the colon cancers and we'll put in some more obvious things that you can't get wrong. And still in 2006, only 6 in 10 labs showed 90% concordance. And finally, colon cancer was removed from the test, and still with 5 different antibodies used, concordance could not be obtained. And in fact, the test was discontinued. So people that do EGFR in their pathology labs today do so without testing, without surveys to qualify the value of their test. And this is, unfortunately, the sad state of what happens in the clinic when antibodies are not carefully validated and carefully used, carefully controlled. The second example I want to tell you about is estrogen receptor. So here's a bunch of estrogen receptor antibodies that are currently on the market and currently used. This one, SP1, is the most popular, and 1D5 used to be the most popular until it was supplanted by this one. The other ones have variable popularity, but the good news is it doesn't look like EGFR. We pretty much can get this one right, regardless of where on the epitope it binds. We pretty much have R-squareds in the 0.9, 0.8, 0.9 range between all these different antibodies, except this one was kind of an outlier, MC20. You can see it's not in this grid. And here's an example of an antibody that was sold by a company as an estrogen receptor antibody, but that doesn't bind to estrogen. It binds to estrogen receptor, but a lot of other stuff. So here's the Western blot, looks pretty good, but a little longer exposure time shows that it binds lots of other things. And if you take an established estrogen receptor antibody where you know and a bunch of cell lines with progressively increased amounts, you can see that in fact this antibody binds to something else more strongly than it binds to estrogen receptor, and led to a whole field of data on estrogen receptor signaling in the cytoplasm. Now that probably really happens, but much of the data in that field that showed cytoplasmic activity was based on the MC20 antibody, which we know binds to something not estrogen receptor. Here's a progressive expression of estrogen receptor in a progressive expression cell line, and you can see that high-level expression is seen by SP1, whereas no expression is not. In MC20, in this exact same cell line, you can see it's got strong expression all the way across. So clearly an antibody that's an example of failure of validation, but that's still published in many papers. So estrogen receptor problems go on. Now, even with the good antibodies like SP1 and 1D5, there are guidelines. The College of American Pathologists, again, now with the American Society of Clinical Oncology, set up guidelines for how we should read estrogen receptor. And the way we read it in a pathology lab is just say any reactivity, and then we estimate the percentage of cells that are reactive. And anytime there's more than 1% of cells reactive, we call that estrogen receptor positive. And if we don't see that, it's estrogen receptor negative, and then we prescribe tamoxifen or an aromatase inhibitor, much less expensive drugs, but still very important for patient care and dramatically improve outcome in breast cancer. However, we don't standardize the threshold for any immunoreactivity. So here's an example of a case that's exactly the same as this case, only this one's stained with, is counterstained with hematoxylin, which is the way we actually look at them all in the pathology lab, and it completely obscures the staining pattern that's seen. So any immunoreactivity is dangerous, and the result of this is different labs, even though they are now these are all CLIA certified labs doing the test according to pathology guidelines, and still have discordances of between 15 and 30% between these different labs. And in fact, you can see here 18% of the cases that were called positive in lab 2 were negative in lab 3, but they show similar outcome, which means that they were actually similar to double positives, meaning they were false negatives. So it's not just a problem with the antibody validation, but even with a validated antibody, a problem with how antibodies are read and used in clinical laboratories. And then the last one is a really simple problem, but I think happens way more than we know, is that antibodies aren't validated at all. And this is from a pathology group where they bought an antibody, and on the tube it said anti-PD-L1. So they proceeded to stain a whole population of patients and drew the conclusion that PD-L1 is associated with poor outcome in human breast cancer. This is what their figure 1 looked like. Now, PD-L1 doesn't bind in the nucleus. It's a ligand that binds the membrane, or that's found in the membrane of epithelial cells. But even their own figures show predominantly nuclear staining, which basically, in my mind, should invalidate the antibody. And we'll talk about all the steps now of how we actually go through validation so that we can have some degree of confidence that what we're doing is scientifically reproducible and robust. So the validation steps are sort of listed here, and I'll go through these. Back in 2010, Jen Bordeaux in my lab came up with an algorithm. She was one of the students who was a victim of the melanoma issue and also worked on the estrogen receptor problem a little bit. And so we decided, okay, we need to come up with a way to validate antibodies so this never happens again. And we came up with this algorithm, which is kind of way too complicated and too hard to remember, but it was downloaded over 27,000 times. So I think at least people are paying attention a little bit. We've simplified it to this now. Is pretty much you need to validate antibodies in the context in which you're going to use them. And so I'm going to talk to you about validation for immunohistochemistry because that's the context in which pathologists use them. These validation steps don't necessarily apply if you're going to do Western blots or flow cytometry or immunoprecipitation or other uses. And I think ultimately when our groups gather to provide guidelines for antibody usage, each validation will be usage specific. So sensitivity, test the signal to noise on TMAs with multiple titers, and I'll show you what I mean by that. So for sensitivity, does the signal localize to the correct biological compartment? That's kind of the easiest thing. You start out, you look at a quick stain and say, is this where I expect it to be? Because if it's not, you can pretty much move on. But then I would use a quantitative method to optimize for signal to noise. Human estimation will fail. So our first step is usually to just look at where it is, and obviously this one was mislocalized, so that's an easy one to eliminate. But then the next step is harder, to actually measure it. So most people that use antibodies in immunohistochemistry are not the same people that use them in immunoprecipitation or Western blot. In fact, immunohistochemistry is often a discipline of pathologists, or the people that do that just do it by estimation because it's really hard to measure stuff in slides. But a number of companies now have produced quantitative measurement platforms. So these are, I'm probably missing some here, but these are all mechanisms that you can actually measure the amount of protein present on the slide as opposed to just guess. Or don't say 0, 1, 2, 3. Actually try to measure it. And the reason is here. That is, there's a difference between counting and measuring. So here I've made an artificial specimen where I've got some spots with 5%, some with 15%, and some with 35% intensity. If I was going to define 5% intensity as positive, and that's my threshold, then this case would be 70% positive. But if it was 15%, it would be 50%, or 35%, only 30%. This is like a qualitative scoring system that will absolutely fail if you try to estimate this by eye, whereas if you actually measure it, you'll be measuring and getting 145 intensity units because you've gotten the total amount of intensity. And this seems straightforward, but this is a really critical point that to this day, 95% of pathologists don't understand. And I don't know about the rest of the world so much, but I think this is an absolutely critical point that you can't count them without knowing the threshold. So you choose a threshold, and in pathology we usually choose the threshold of the limit of detection of the test, which isn't the greatest threshold in some biological circumstances, but in fact can at least give you more information. So in summary, there's more information content in measuring than counting. Counting is variable in the real world due to inherent threshold variabilities, and H-scores, which try to be a pathologist's approach or a human approach to estimating intensity, where you have intensity times area, has been attempted but failed. And in fact, probably the METMAB trial failed on the basis of the inability of pathologists to reproduce H-scores. So this is just an example of what happens when pathologists try to do this, is these are H-scores on this axis or on this axis, and this is actually met. And so we're measuring the protein met. And you can see when a pathologist estimates, they don't say it's 23 or 27%, because they know they can't reproduce that. But they think they can reproduce 30 versus 40% versus 50%. So you can see this sort of incremental estimates, and you can see the breadth within an estimate. For example, 100% by the pathologist, or an H-score of 300, actually ranges across about a 50% dynamic range of estrogen receptor. And this makes a difference. This is the survival curve by H-score. This is survival curve by quantitative measurement of quantitative immunofluorescence, where you actually can see a difference that's not visible by an H-score. So this is a method I'm going to talk to you about, because this is a method we use in our lab. I don't necessarily think this is the only method, but the idea, and when I talk about a quantitative immunofluorescence score, an aqua score, this is what I'm talking about. Basically, it's the sum of the intensity of the compartment pixels. That is, if we're using estrogen receptor as an example, what is the sum in this field of view of the intensity of all the pixels divided by the compartment pixel area? So all the DAPI positive pixels will be my denominator. And so you can get a numerator over a denominator which simulates a concentration to the best of our ability to generate a concentration. And many of the previous softwares that and companies are aware of this, and they can either do this or they have some alternative counting method if they don't want to try to do a concentration. This is an example of another method we use in our lab. This is actually an FDA-approved method for measuring estrogen receptor, and it takes a continuous score and converts them into a 1, 2, 3 scale so that it's more familiar to pathologists, but it's also a method for measuring protein on slides, although it's been essentially limited to a 3-category test. We generally use fluorescence for this region. This is HER2, 2 different antibodies to HER2, one cytoplasmic domain, one extracellular. And you can see that when you measure them with DAB compared to quantitative fluorescence, you don't appreciate the full dynamic range with either antibody, so showing the sort of the plateau. And that has to do with just the physics of measuring absorbance versus emission. And so we generally try to measure everything with quantitative immunofluorescence. I want to show you another example of a problem in measuring HER2 of being unable to detect the differences. These are, this is where Herceptin binds here, here's where pertuzumab binds here, and this is the HER2 molecule with two, this is a well-known antibody that's used in about 6% of labs now, and this is the lab is about 1%, this has about 45%, and maybe this has about 25% market share. These are the antibodies that they test, and they do a test that's a DAB-based immunohistochemical test, and on the basis of that test, pretty much prescribe $100,000 worth of drug. And so are these different? They bind to different domains. How can they all be the same? This one actually is not used very much in the clinic. So a group in the Netherlands, led by a breast cancer pathologist, Mark van der Wijver, looked at SP3, 4B5, and the Hercept test, and asked the question, when we compare them, is there any difference? And without looking in detail in the table, you can look down here. Basically, the discordance between the extracellular domain and the cytoplasmic domain, or two cytoplasmic domain antibodies, was less than 1%. But that kind of defeats biology a little bit, since we know that the HER2 molecule is processed. And in fact, if you use a quantitative test, quantitative fluorescence, you can see here's the extracellular domain, here's the cytoplasmic domain, and a good number of cases don't have any extracellular domain. It's been cleaved off. This is not news to anybody because that's how HER2 works. That's the biology. But we can't tell the difference in our assay amongst those two things. And so even though if we look at some of these cases, for example, this case, the extracellular domain looks equal to the cytoplasmic domain, but this case, you can see lots of cytoplasmic domain, but no extracellular domain. And here's the examples of that, so you can really see that this exists. And in fact, it can even be shown with DAB, except that a pathologist might call these cases equivalently because there's full cytoplasmic staining, and even though this has a greater intensity, you have reached a threshold here, and that's probably why they're different. I won't go into the details, this has all been published, but it actually affects outcome, and we're working on trying to prove whether or not this is important in response to therapy. So in summary, with sensitivity, does the signal localize to the right place? Use a quantitative mechanism to estimate signal to noise, and we'll get into that now. So how do you actually estimate signal to noise if you're going to measure an antibody? And so this is what we do. We use concentrations of the antibodies or dilutions of the antibodies. With monoclonals, we actually try to use concentrations, but that's on another graph. And you can see that you can take a series of cases. Now, these are not, these are actually small bits of patient cases, so we can accommodate the full dynamic range that we know are negative, and those are the bottom 10%, or that we know are positive, and those are the top 10%. And then you can generate a signal-to-noise ratio on actual human tissue. And so you can see where the optimal titer was is where you optimize your signal-to-noise ratio. And in fact, this is the same lot of antibody from the same vendor. And you can see that actually it changes a little bit over time, just with variables that are in the lab. So we actually repeat this signal-to-noise ratio every time we use the antibody fresh, because there are small variables in the system that we can't control. When you look at the signal-noise ratio, you can actually confirm this, or we have been able to confirm this with mass spec. And I think this is an interesting, if you look at the scatter here, as you get, so what we've done is measure with a collaborating company the number of attomoles per microgram on the slide, using mass spec on the slide. This is something that a company called Oncoplex DX does. So you can cut your tissue onto a slide, then they'll basically take it off by an ion beam, and then suck it off the slide and measure the exact amount. So it provides a standard for us to try to test against quantitative fluorescence, and we're in the process of doing that now. And what you can see is that when we have the titer right, that is, if we've optimized the titer, we get really good agreement with the true value according to mass spec. But when we don't have the titer right, we get much more scatter. And that's exactly what you'd expect, which shows you the importance, I think, of measuring the quantitative fluorescence and using the correct titer of the antibody, having the sensitivity correct. So this is actually measuring signal-to-noise using cell lines, but you can see that in these PD-L1 antibodies, one of them in particular has a much greater signal-to-noise in actual patient specimens compared to two others that I'll talk about in a minute. So the next thing is specificity. If you're going to validate an antibody, you should make sure it's specific. So Western blot's a good start, but it's not sufficient. We either use progressive expression of cell lines or overexpression of the molecule of interest, the target, and negative cell lines, or sometimes knockdowns or knockouts, and this was mentioned a little bit. And then the other thing that we've started doing is confirmation of reproducibility with a second antibody. Because if an antibody cross-reacts, it's unlikely that a second similar antibody will cross-react with the same pattern. So that can also be another solution for proving antibody specificity. So this is the example of estrogen receptor where we transfect in using a inducible vector system. So more DOX, more estrogen receptor, and you can actually see that the antibody expression level and the quantitative fluorescent level goes up. So that's a good validation for an antibody. And you can actually see all your negative cases and then your positive cases, so you can even find a threshold and actually measure a threshold this way. The other thing to do is a simple knockdown experiment, which is often done with shRNA or siRNA. And if you take a cell line that has nice expression of your protein of interest and then knock it down and see the staining go away, that's pretty good evidence for specificity. It's not perfect because it's a cell line. It's not the tissue in which you're ultimately going to use the test. And for that, I want to talk about what's going on in PD-L1 for a minute, because this is an area that's become very important in the immune therapy world. And one of these drugs, that is this one, pembrolizumab, this antibody test on this platform is actually required for prescription of the drug, whereas this drug is prescribable in the absence of a companion diagnostic test, even though they do have an FDA-approved companion diagnostic test. But there are 4 different antibodies that all are carefully validated to bind to PD-L1, theoretically. Here's a bunch of antibodies that we tested in our lab that do not validate. And so some of them had no signal, which may have been an issue, but what was most common is that we found that there was non-specific binding in non-expressing cell lines or in other tissue. That is, that it's not that they didn't bind PD-L1, it's what else that they bound or how they cross-react that was really the biggest problem. And cross-reactivity can be a huge problem in situ because you don't see it. It's very subtle to see. And in fact, we made this PD-L1 index array to test this, where these are 30 cases of lung cancer with different ranges of PD-L1 expression, and then a whole series of cell lines and some placenta and tonsil. And we use this TMA to look to compare these different antibodies. So here's E1L3N, SP142, which is a Ventana antibody. This is a cell signaling antibody, and this is a spring antibody. And you can see that the antibodies overall compare fairly well, but this is the tumors where there's some heterogeneity, and this is the cell lines. So the cell lines are much safer to compare the antibodies in terms of cross-reactivity, or in terms of just pure binding of the antibodies, but in cross-reactivity there may be more diversity, or this may actually represent heterogeneity of the specimens. But the reason I show you this is we found one antibody, E1J2J, that is an outlier, and I think this is cross-reactivity, but the problem with this cross-reactivity is we had no previous way to discover it other than to look at a bunch of cases and look at multiple antibodies. And so this is a type of specificity, a specificity problem that's extremely hard to detect because you can only do it when you actually test it in a series of patients of which you might ultimately use the test. So specificity failure, in general, omission of the primary antibody does not prove specificity. Peptide competition does not prove specificity. It's most often not the absence of target binding, but rather target binding and also cross-reactivity, which can be hard to find. And it may only be revealed when the antibody is used in the application in which it was intended. So finally, the last category in the last 5 minutes is the issue of reproducibility. And we try to get reproducibility with a complementary reagent, different days, and then ultimately potentially reproducibility generated, as Andrew suggested, by sequencing of the binder. We have not yet achieved that. This is just an example of two different antibodies, both extracellular domain antibodies. Gives us high degree of confidence that our assay is in fact correct with this kind of reproducibility. And if you're quantitating it, in fact, you can calculate a CV. So you calculate your coefficient of variations, different days, between different days, different operators, et cetera. And if your coefficient of variation is below 10%, that's where you should be with an ELISA assay. That's where you should be with immunohistochemistry. This was actually showing the lack of reproducibility you get automatically. These are actually two different platforms. So this one's run on a DACO autostainer, and this one's run on a Discovery Ventana autostainer. And you can see there's clearly a lack of reproducibility between the stainers. So you can't rely on a machine to do this for you. You actually have to test it yourself. And actually have to, in order to find this, you have to measure it. If you just look at it, these will all look fine. So finally, for a summary sort of QC is test sensitivity with a cell line series, test on a non-expressing cell line transfecting with a high or expressing cell line with and without knockdown, and then test for reproducibility and consider a second antibody. And so with that, I'll just briefly talk about who's responsible, and the answer is I don't know. We see some people actually have validation services and validate their antibodies. Other people, if you Google their website for validation, it says no records can be found that match that search criteria. Perhaps more recently, there's a number of people that are actually validating antibodies or having external validation of antibodies. Some of the vendors, quoting cell signaling, Craig Thompson said in a webinar that I participated in that it is the responsibility of the company that produces the antibody to validate the product, which I really like to see. I'm not sure that every company agrees with him, but it's interesting that Abcam actually moved to provide knockout validation for its antibodies. So at least there's beginning to be some kind of movement toward a responsibility on the part of the vendor to at least take up some aspects of the validation. So I won't go through I've kind of summarized this before. For scientific use of antibodies, which I understand is about 1/4 of the market for antibodies compared to diagnostic use of the antibodies, although I'm looking for that statistic if anyone in the audience knows it. Except for pilot experiments, we pretty much stick to monoclonal or recombinant antibodies. For IHC, use quantitative tools, and immunofluorescence is much more accurate than chromogenic tools. Do a quantitative titer to optimize signal to noise and validate the antibody as discussed. And also considered paired antibodies. If you have two antibodies that are at appropriate adjacent epitopes, you can have a lot more confidence in your result than you might get from a single antibody that has the ability to cross-react. In the clinical setting, there's also this paper by Fitzgibbons et al., which defines what CLIA labs should use to validate their antibodies in terms of the number of tests they should test. We've also introduced the idea of TMA standardizations where you put in a TMA with every stainer run, and that TMA is like the one I showed you for PDL1. There's now a company called Horizon Discovery that's making these TMAs commercially available so that they can be run routinely in clinical labs in order to have a sort of every assay test of the antibody validation. Right now, there are no clinical antibodies that are truly quantitative. Only ER, HER2, Ki67 are semi-quantitative, but I think, and PDL1 is now semi-quantitative. But I think the reason for this is we've been inappropriate in the way we've measured antibodies in the past, and it will be valuable at some point to introduce quantitative use into the clinical sphere. So this is my last slide. I always like to acknowledge the group, my lab group that does all this work that I show, and the granting agencies that I had mentioned. Are there any questions? Thank you.
Very nice talk. I'm Scott Muirhoff from Abbott Diagnostics. I'm the Director of Biologics Discovery. Just a comment on the topic of validation. It's really impossible to validate an antibody for any given application. And I would never trust a vendor to do my validation for me. The validation is really the responsibility of the end user. We have cases where we have actually products on the market where we have a single antibody that's been characterized to the 5th decimal point. And we know that we can use it in one assay as a conjugate. In another assay, it has to be used on the solid phase. It hits the same target. We've sequenced it. We know it very well, but we have to validate it for each and every use. So even if a vendor says that they validated something for an ELISA or Western blot, you've got to do it yourself, and you've got to do it 10 different ways.
So that's a great point, and I very much agree with that. In fact, a previous, I think vendors have a responsibility as well to provide an antibody, but then you still have the end user has to validate it. The problem is that that doesn't happen. And in the pathology litter especially, most pathologists don't actually run the validation aspect of their lab. They come up with the project, but they don't actually run the validation aspects of their lab. So there are barriers to validation. Another issue arose is that if you're a vendor and you sell 18,000 antibodies, it's unlikely you'll be able to spend the $5,000 to $10,000 it cost for each of the validation studies that I did. And so there's a balance between how this, who carries the responsibility.
Hi, excellent talk. My name is Tintam Jaratat. I'm from Morphosis in Germany, and I happen to be the only pathologist here in the audience. Yeah, many valid points. I have a specific question. Actually, in my past, I did much immunohistochemistry in the therapeutic area, and there was increased interest in detecting by IHC ligands, such as heregulin for HER2 and HER3, or amphiregulin for EGFR, HGF for the MET area. How do you approach this signal-to-noise ratio in tissue with regard to ligands? Because you usually have some kind of a blush in the tissue where you aren't sure is that noise or is it ligand that's around in the microenvironment.
Right, so you've brought up a really interesting point, is that another key problem with immunohistochemistry is the protein has to be fixed there. And for most ligands, that's not the case. That is, for HGF or amphiregulin or many of the ligands, the amount of ligand present has to do with pre-analytic variables. That is, variables that occurred between the time the specimen was taken out of the patient and the time that it got into the processor. And so, in general, we don't try to measure ligands at all, but rather receptors, which we can more reliably count on being present at the time of fixation and not being lost in the process of solubilization and formalin infiltration or formalin fixation and subsequent tissue extraction and paraffin infiltration.
I have a rather financial question. So you did a lot of validation yourself. So obviously, you have to use the same batch that you use for validation in your real assay.
Yes.
So can you give a rough number? How much more than you actually need you have to buy this antibody?
So
What
that's
what
a
part
great
goes
question
to
because
an evaluation or validation, in which part you actually use? It's one question. And second, if your validation study find out that this antibody are not acceptable for whatever you're doing, do you ask the vendor to reimburse you, or it's just lost money?
unfortunately, it's a loss, and because we don't want the vendor to, if we find out the antibody doesn't validate, it cross-reacts, we don't need any more of that antibody. It's never going to, if it doesn't validate once, the next lot is no more likely to validate. And so generally, we move on
Right,
to another vendor.
But if the vendor doesn't carry any financial burden, then he's not penalized for bad antibody.
That's true. Unfortunately, that's true. And that's what, so obviously we need some other system to solve that problem. And I would argue that to answer your first question, that is, how much do we need? And the answer is it shouldn't make any difference. We need however much we're going to use. And then when we buy the next lot, we validate it again. So that it would be great if we didn't have to validate the next lot, but if it really is binding to the protein and it's not just an assay, but we're actually measuring protein on a slide, we have to repeat that validation. So we kind of set up these index arrays that have knockdowns and overexpressors or a range of cell lines in them so that our validation can be much more efficient on the second pass, especially if we know we're going to need to validate a number of antibodies or validate the same antibody numerous times. So you can try to make it more high throughput.
Right, but what you currently have, 30%, or 20% goes to validation of molecules?
There's probably less than half validate.
Less than half, but close to half.
Yeah, maybe. Depends on the. It depends. So a lot of so without trying to offend anyone in the audience, especially vendors, there are some vendors we never use. There are some vendors we always go to first. If they have the antibody, we go because we know that their validation methods are the same as ours. And so we have, when we buy an antibody from that company, we feel like we have a 95% chance that it's going to validate. But if they don't have it and we have to kind of go down our list, there's some companies on our list where if we'd be happy with 50-50.
Hi. I would like to ask a question on behalf actually of a patient. If you are a patient and you have some type of diagnostic based on pathology, how can you really assure that your diagnostic is right, and what can you do?
Yeah, that's a real problem, is that how do you know that it's right? If you're a patient, how do you know that you're going to respond to trastuzumab? Well, that's the standard of care. And so send your specimen to a CLIA lab that's certified, and that's the best we can offer right now. Unfortunately, that means we can't distinguish between extracellular domain and cytoplasmic domain. That's down the road. But someday we will be able to, and that will probably make it into a CLIA lab. Right now, if you don't have any extracellular domain, you're not likely to respond to trastuzumab, but there's currently no way to find that out in CLIA-certified labs. However, having said that, if you want to know whether you're estrogen receptor positive or negative, you probably have about a 90%, 85 to 90% accuracy, which is about as good as we can get for that lab test.